I'm just going to start giving some framework and expectation to people who are with us today. So for those who don't know, this is a second talk on hyperscanning that we are organizing. A couple of weeks ago, we had Suzanne Dicker from NYC and Martin Imhoff and Carl Philipp Flusch from University of Constance, and we kind of gave a bit of tips and tricks about hyperscanning studies, but we saw that a lot of people actually want to hear more about it, and we also got a pretty useful feedback on several issues, and we hope that we are going to address it today. So we added the Getting Technical into the subtitle, and I hope this is going to give us the chance to do exactly that. I'll be your host. As I said to Francisco earlier, I had to put my picture so that you see that usually I'm prettier than today. And some useful tips before we start. Goal of this webinar, I cannot help but remember my PhD days, and I was always suffering because I didn't find any material between the research papers and the books. So in research papers, everything was very specific to address the certain issue described in the study, while the books were too broad and too general. And sometimes, a lot of times actually, I had a problem of getting the scope of something in my mind before I dive deeper. That's one of my personal goals with these talks, to sort of give an overview and then go into a few specifics that we identify as relevant, and start from there. We are going to have around 90 minutes of time, where we have actually 1 hour reserved and 30 minutes optional, depending on the questions that we get. Please feel free to send your questions. There's an option to send questions. I promise to read as much as I can, although please excuse me if I don't answer. Actually, if I don't put out all of them, this is because I'm not that good in multitasking. So, but I hope that it will be useful anyway, and we will record this video, put it out there on YouTube. There's a Brain Train channel there, and you can look it up. So this is a bit of agenda for today. We're going to have the talk that I just introduced. So we just crossed the first item of the list. We are going to have Martin and Francisco giving a short talks on what they do. It's very interesting, and they are both in this domain of hyperscanning, and in broader terms, in very much technical about EEG, with a lot of experience. And then we are going to have a discussion and answering questions, and then we are going to close down, of course. So Martin Bleichner, I know for quite some time now. Martin, can you remember for how many years? 7, 8?
I think 7, yes.
All right. And Martin is a real pioneer in mobile EEG, and it's really hard to say in a few words, but he tries to bring the EEG more to the outside world, to the real life environments, and adopt both the environment and the EEG to these new conditions. He's also one of the pioneers of this new modality of EEG recordings called C-grids, where you actually record from a small number of locations around the ears to make it, to conceal it in a way, so that one day we can bring it out of the lab and hopefully in real life for the betterment of all of us. And I see that in his description, he likes to say that he focuses on negative consequences of annoying sounds. Maybe that's somehow related that he is in Germany, and maybe Francisco wants to focus on positive effects of sounds. So this is about Martin. I'm really glad that that he will tell us more in a bit. And he just has published his study on word-to-word interaction with the two participants, which is a hyperscanning study and very interesting concepts on engaging, actually making sure that that the participants are engaged on a biological level, and then using that to further explore synchrony. Francisco Parada, I know for considerably less time than Martin, but I feel somehow that I know you longer. So we met last year in Belgrade on the workshop on Mobile EEG, and he's the director of the lab at the Universität Diego Portales. I don't know, my Spanish is not that good.
That's okay. That's okay.
In Chile, and he used to work in Harvard, and it's on some very interesting stuff related also to brain rhythms, synchrony, interaction, and how our brain actually converts the inputs that we have into actionable results, how it filters it, how it chooses, and how this turns into action. And I think that we are broadly in a small closed circle of people who know each other, and I'm really glad about that. So without further, oh no, I was just about to start early. I have a small request for everyone. So we have a small poll to just see where we stand with the overall knowledge of hyperscanning, and I'm just going to share the poll with you, and ask everyone to vote. So I'm going to launch the poll and also allow the participants to answer this. The question is, how difficult do you find setting up hyperscanning EEG study? Okay, we have one vote so far. Okay, now it's building up. All right, interesting. So, okay, some people are still voting, and I think we're done. I'm going to share the results. So we have actually a majority of people that have pieces of the puzzle, but not sure how to do, how to put everything under control. And that's actually very good news for us today, because this was the very motivation of being here today. I'm going to go back to the slides and actually give word to Martin. So Martin, please proceed.
Yes, hello everyone. We just see that I can share my screen. Can you see my screen? Yes, okay. But we just, before we started with the online version of this, we discussed how strange it is that we are now giving presentations and we don't have any feedback at all. So my goal in research is that I want to understand social interaction. I want to leave the lab to go to natural environments, and now I'm going to give a talk without any social feedback. So all my jokes will just enter the internet and get lost in the end. So it's a really strange feeling for us here in this very strange time. So my name is Martin Bleichner. I'm from the University of Oldenburg, and I'm head of the Neurophysiology Everyday Life group, as Ivan already introduced myself. We work primarily on mobile EEG for beyond the lab brain recordings to study auditory attention in real life. Our goal is to leave the lab and to study brain function in everyday contexts. For this, we will develop the technical and methodological tools. But today, I will not talk about our work on EEG, but in the technical aspects of studying social interactions with simultaneous mobile EEG. So in a project in collaboration with the Technical University in Dresden, we did a series of studies to understand the neural processes underlying improvisational theater. The project was called Impact, Improving Patterns of Social Interaction. All of this work was actually done by my former PhD student, Tatjana Gorigliat-Fellingsdal, as part of her PhD. If you want to dig deeper into these topics, you can have a look at a sequence of publications we published about this project, which are shown here on the slide. Improvisational theater, for those of you who don't know it, is a form of theater where there is no script. Instead, a scene develops from scratch. It emerges between the actors. They make it up as they go, by reacting to what the other person just said and by adding something themselves. In order to have a coherent scene, the actors have to anticipate, they have to predict what the other actor is about to say. On the other hand, they need to react flexibly if their expectations are not met. The interesting part for us was to see how these expectations develop during such a scene and in how far we can use EEG to look into the underlying neural processes. And I will quickly go over the study design before we go into the technical part of it. So when we started to develop this idea for this project, we thought we could use simply mobile EEG to study the actors on stage. But it became pretty clear pretty soon that this would be an experimental nightmare. We simply would not know how to relate the EEG recording to anything that happens on the stage. What part of the EEG would actually look like? How would you determine if an event is expected or unexpected? So we went on a journey somewhere between a natural interaction and sufficient experimental control. And I think this is crucial for any kind of social interaction experiment. So we took a specific game from improvisational theater, the so-called word-by-word game, sometimes also called one word at a time. In this game, two actors play one person and they take turns in saying the next word, but they don't know anything about what the other person is about to say. So, for example, we have two persons here, and the first one says, I, the other says, want to buy a hamster. And this is something that emerges from the participants, and they have to react to what the other person says. And if people play this game, some people produce fluent sentences, and others fail miserably at doing this task. And we wanted to know what's underlying this. The nice thing about this game is that it lends itself as a paradigm for EEG studies. It has a very clear temporal structure, and therefore it allows to do ERP responses. On the other hand, it allows for controlled experimental manipulations. If you tell the green guy beforehand that they want to buy something in the bookstore, and the blue guy that they want to buy something in the pet store, you know that at the time the word hamster is uttered, that one guy is very surprised about this word. And then we can study the brain response to this word and also the reaction of this guy towards the word. So he might not be able to continue the next sentence because he's so surprised and so flabbergasted. So what we did, we took this setup and we placed two people into our lab. Both were equipped with 24-channel mobile EEG systems. Both were in front of their own computer screen, and we recorded from both of them a microphone that was placed in front of them. On the screen, they got instructions, and then they took turns in saying the words of a sentence. We designed the task in such a way that sometimes the words the other person uttered matched the expectations, and sometimes they did not match the expectations. So the paradigm looked like this. We recorded two EEG streams and two microphone streams. We wanted to know how does the brain of the green person reacts to the utterance of the blue person and the other way around. For this, we used the audio signal of the one person to epoch the EEG signal of the other person, which makes it necessary to record these different signals, the EEG and the audio, in very good temporal precision together. Before I go into the technical details, I would like to mention that we were actually able to find the expected ERP effects So for the unexpected utterances of the other person, or the unexpected utterance of the other person are processed differently from the expected utterances, which is reflected here in this difference in the N400 response. And also the underlying topographies give a further indication that we actually have useful data here and that the paradigm as a whole worked. So we can conclude that we can actually measure mobile EEG simultaneously and to study two people at the same time. And now my presentation is running on its own. So much for data synchronization. So now I would like to go deeper into the technical aspects of recording EEG from several people at the same time. And this brings us to the topic of data synchronization. You might know from some old movies where they have some important joint objective that they always raise their watches and compare their watches and synchronize them so that they follow all the same timeline. And the same is true for any kind of data recording. You need to have a common timeline for all your recordings to relate them to each other. The important bit here also is that this synchronization needs to be done regularly. If those two gentlemen would meet the next day, they would again have to synchronize their watches because over time their watches start to drift apart. If you're not into movies, but you're more from the YouTube generation, you know this phenomenon from some videos where the sound and the audio drift apart. This is very annoying when you have it. The person in the video starts moving the mouth, but the sound comes later. So this is also a problem which you observe when you record several data streams at the same time. Next, I will introduce a couple of basic concepts in this regard and then move to how we solve that in our study. So the goal of EEG studies is to relate the EEG activity to something else. So this could be an external event. You present a sound or a picture to a participant and repeatedly record the brain's reaction to it. This could also be an internal event where people are engaged in some mental activity, for example, for brain-computer interfaces, when people are asked to imagine a movement. Or that you want to relate the brain activity of one person to the brain activity of another person. Depending on your research question, you need more or less accurate timing precision. If you are interested in the boredom of a group while watching a movie, or how engaged the audience is while listening to a live music performance, you may be interested in larger timescales, effects that unfold over several minutes. For such a question, you can live with relatively poor timing. You might want to say something like the alpha activity in the first half of the movie is lower than in the second part of the movie. If that is your objective, you can live with quite some temporal unprecision. However, if you're interested in the interbrain synchrony of two people that are engaged in a conversation, you may need a much higher temporal precision. I once had a colleague who could not replicate the results of his own study. The peak latency of his ERP was off by 5 milliseconds. And he was really concerned about this for quite some time. And in the end, he figured out that he did the first experiment with headphones on and the second experiment with speakers. And the difference in the peak latencies could be explained by the velocity of sound and the distance of the speaker to the ear of the participant. And if that's your level of precision, then of course you have to be very accurate in what you're doing. So try to have the best timing possible for whatever you do, but also keep in mind what your research question is about and what kind of inaccuracy you are willing to accept. I would like to continue with some basic timing concepts that are important to understand. First of all, offset and jitter. So let's say you have some auditory stimulus to which you have some basic brain response. In your experimental setup, you record the brain activity, that is the brain response to your stimulus, plus some noise and some trigger event, which is indicated here in red. So you play a sound, the brain reacts, and you record that. You do that in an experiment several times. And in the ideal case, the triggers in the EEG are perfectly aligned. Whenever a sound is presented, the trigger is written reliably. When you then epoch your data, all events are nicely aligned, and you can average over all trials, and you get your brain response, you get your ERP. This is what you should have for most lab-based systems, basically. In such a case, you can say something about the timing of your brain response. You can say something like the N100 response is exactly 100 milliseconds after the stimulus. And the ideal case would simply look like this. All the trials are nicely aligned. All the brain responses are nicely aligned. However, what you sometimes observe, especially when you work with wireless systems, is that there's some kind of offset. One of your systems is ahead of the other. In this example, the brain response appears to come late relative to the trigger event. This can have several reasons. The trigger is written too early, or the sound is played late, or the EEG signal lags behind because it takes some time to transmit the signal from a mobile device. So those are all possibilities. If this constant offset, if there is a constant offset in your system, that is, one system always lags behind the other system in the same way, you can quantify the system, this offset, and correct for it. That's actually fine. If you average over your trials, you will still get the right brain response. Much more problematic is when your system has a variable offset. And that also happens quite often. When there's some kind of so-called temporal jitter. That is, your system writes some trigger, but the sound is actually played at a different time point. One time the sound is played exactly when the trigger is written. Sometimes the trigger comes 24 milliseconds too early, sometimes 130 milliseconds too late. This is actually something you observe very often on Android phones. They are notorious for this kind of behavior. You tell the phone to play a sound and the phone to send a trigger, and the phone then plays the sound at some point and plays the trigger at some point, but this can have huge differences. And in the end, if you would average over these very jittered responses, you might actually average out your effect altogether and don't have anything but noise in your signal. So this is really a nasty problem, which is very difficult to get rid of afterwards, so try to do it beforehand. The next important concept is drift. As I mentioned that when you see a video where the video and the sound drift apart, the same can happen in EEG recordings. At the beginning of your recordings, you are still synchronized. At the end of the recordings, things drift apart. So in the example I show here, you have in the beginning the sound is sent and the trigger is sent at the same time. And then slowly this trigger signal shifts relative to the onset of the sound. And what you would have observed in the end is that your signal drifts over your epochs, and in the end, again, if you average things, you don't get anything useful anymore. The problem here is that one clock runs faster than the other. Also quite common to see. Important for you is to know that all these effects can happen. There are numerous reasons why they happen, and they will happen to you. So you have to know about them, and you have to be careful to try to avoid them and do everything that you can. So this brings me to my last concept, which I would like to introduce, which is the sampling rate. The sampling rate, often given in hertz, indicates how many samples a device records in 1 second. In our studies, we had 2 EEG systems sampled at 500 hertz, a microphone sampled at 44/100 hertz, a video at 24 hertz, and some irregular event markers, which indicated when something happened on the screen. So the 500 hertz means that the EEG takes 1 sample every 2 milliseconds, whereas the video takes 1 picture every 41 milliseconds. So the sample rate is important to consider when relating different signals to each other. When you use the video, for example, to detect some event, the opening of the mouth, for example, you may not have sufficient temporal precision to do something with your ERP. Also, a word of caution here, especially with mobile devices, you want to check whether the sampling rate that is indicated on the device is actually correct. This is if you record a 2-second data segment of EEG with a nominal sampling rate of 500 hertz, there should be 1,000 samples. While this may appear trivial, this is not always the case. When I started to work with the smarting amplifiers, actually, they said that the device was recording at 500 hertz. I did some test recordings and realized that sometimes there were more samples and sometimes fewer samples. When I asked the manufacturer about this, they told me I was holding it wrong. I kept on bugging them until finally they sent me a firmware update, and the problem was solved. Afterwards, they told me, we told you that you were holding it wrong, but the problem never occurred afterwards. So don't trust the manufacturers, but be very critical towards the tools you are using. So how can we now record the different signals in a synchronized way? In a way that all the signals have the same time reference and in such a way that we can relate them to each other. If you reply to this with, we just press on start on the several devices at the same time by shouting 1, 2, 3, push, my reply to you would be, do we push on 3 or on push? And my actual reply would be, just don't do that. Whenever we want to record several data streams, also with different sampling rates from different devices, we use a software that is called the lab streaming layer. The streaming layer, LSL, is a system for a unified collection of measurement time series in research experiments that handles both networking, time synchronization, real-time access, as well as a centralized collection, viewing the data and recording the data on disk. That means you can have that means that you can even record devices that are distributed over different computers. LSL is open source and free of charge. It is developed by some very smart people and is immensely useful whenever you want to do synchronized data recordings with mobile devices. I'm not a developer, but just a user. So one key element of LSL is the lab recorder, where you can record several data streams into a single file with a common time frame. Lab streaming layer works in such a way that any device that supports LSL broadcasts its data together with a reliable timestamp. The lab recorder listens to all these different data streams and integrates them into a single file with a common timeline. You can check online whether your devices are LSL compatible. Here's a small list of supported EEG devices and stimulus presentation devices, but there are also eye trackers out there, keyboards, gaming pads, you name it. It's quite likely that whatever you use in research is already supported and is more or less plug and play. With this list of references where you can get LSL, where you can check for devices, and where you can get some tutorials, I would like to conclude my talk here, and I'm happy to answer the questions afterwards.
Okay, Martin, thank you very much. This was really a very, very nice introduction and exactly what we want to do. So we are convinced now that hyperscanning is good and we should proceed to doing those studies, but some technical aspects are rarely known. Time synchronization is one of them, and at least a lot of my colleagues still have to, I believe, catch up with this fact that you introduced, that if you synchronize watches one day, in the second day, they will again need to be synchronized. Okay, it's just to remind everyone that we have Q&A options, and then everybody can actually type questions. So after Francisco says a few words as Martin, we can jump to those questions. So getting back to our agenda, now it's Francisco's word. So Francisco, please be my guest.
All right. Well, thank you for having me over here in my house. But it's going to be fun to actually talk about a little bit more, and actually continuing from what Martin was saying. So I wanted to go and put some further ingredients. Actually, Martin and I didn't plan our talks together, but this is going to be fit really nicely because there are some spots where he already covered, so I will be able to actually just skip those and we can continue. And I'm going to give you even more pitfalls. So let's begin. So first, we're going to ask two questions. First of all, should we even try? Okay, well, that's the first question I think someone that is starting, a grad student or a postdoc, or even a young professor just starting their own lab, they are going to say, should I even try moving away from the classic ERP paradigm or event-related classic paradigm in the laboratory into hyperscanning or more mobile settings? So I'm going to just briefly talk a little bit about that, and then we're going to go into some good practices and pitfalls. So let's go with should we try? This slide partially stolen from Simon Ladouce. He's in the audience somewhere there. So he describes very well these models, how we are moving from the classic input and output models towards something more environmentally friendly, let's say, that considers environment in a little fashion. Okay, so I would say that this is where the regular neuroscientists are here. Martin and his group, Stefan Devener as well, they are of course in the avant-garde movement, but the majority, I would say, of researchers might actually situate themselves over here. However, it's a little bit more, you can get more radical than this. However, it's dangerous to go alone. I don't know if they will understand this reference, but take for recognition, I would say it's dangerous to go alone because it's not only technical, but also you need theory. You need to go with a very clear theoretical background into this huge fight, because it's epistemological at the end, okay? It can be methodological, but also it's epistemology here. So for recognition, quickly, I would say that the for recognition approach basically suggests that cognition is going to be an embodied, embedded, and extended phenomenon that is going to be better studied within an enactive perspective. What all that means, well, we have no time to actually go very deep into that, but on Friday, the 24th or the 25th, I'm going to be giving another webinar about this topic precisely. So I don't know, follow me on Twitter or whatever, and you can find out if you really want to learn a little bit more about for recognition in this setup. However, we're going to be, from this epistemological standpoint, we're going to go a little bit farther away from the neurocentrism where like experience happens only inside the brain, but also is going to be a necessity of adding a more embedded approach where cognition is, of course, produced in the brain. The brain is the organ we should be studying, but that brain is embedded in a body, and that body is embedded in a background, be that natural or built. And that's very, very interesting because it changes how we will ask our questions. This has set up, and again, taken from Simon's very nice review from 2017. This has basically driven us into laboratory setups and mobile setups nowadays, and we call this mobile brain body imaging setup. This is very related because basically hyperscanning can happen in both these situations, and your questions actually will drive the need or will drive the necessity of actually doing it in a more laboratory based or in a real world based setup. That's going to be for you to decide. So again, it's dangerous to go alone. Take mobile brain body imaging. That's another tool that we're going to actually be needing to move towards these more avant-garde research agendas. So again, Simon, thank you for your slides. We are moving away from the sensory input-output models and going into a more radical embodiment, or what I call the 4E. It's not my term, but I've been using it. So where those models are going to drive our questions. And one of the questions we do is like basically, what do humans actually do in the real world, so we can actually go and study that? And one answer that I like to give all the time is social interaction. The environment is mostly composed by conspecifics, and we're all the time doing that. This is from a long time past, because now we only do Zoom meetings, but still, the social interaction is still there. So from the original invitation that Klaus Graman were doing in these setups where people were moving freely in the laboratory, it seems necessary that we need to do a next step. And the next step is actually joining with some other person in the laboratory or in the real world. So here slides taken from Artur Czasumski. He's also in the audience right now. A very beautiful review he just published. Check it out if you haven't. On hyperscanning, if EEG mostly, and why would you even do fMRI? Let's just take it out of there. So in EEG, these setups, you know, they are very complicated, and this is apparently, or at least what I think, is the way to go. So hyperscanning and mobile brain body imaging, I think they are too compatible and too further down the road goals that we definitely should address as one. So against the question, should we even try? The answer would be hell yes. And so let's go into some good practices and pitfalls. So basic EEG pitfalls 101. Everything that can go wrong in a regular vanilla flavor ERP task in your laboratory can go absolutely wrong in your hyperscanning setup, in your mobile setup. So basically, set up the equipment as recommended by manufacturers. I completely agree with Martin, even though you don't have to blindly trust your manufacturers. Sorry, Ivan, but you don't have to do that. Partner with them, work with them, but also be suspicious. Know your equipment for sure. So make sure that your equipment is set up correctly. The impedances as well, that trust your manufacturer. If your manufacturer is telling you that you have high impedances as your system, go for that. If you have to go lower, go lower. So follow those instructions for sure. Disconnect participants from other electrical devices if possible. Because I remember in the laboratory, in the regular, if it's possible, of course, if they are like in a hospital, like with a respirator, don't do that. So basically, phones or any other equipment that might actually introduce another signal, we usually recommend to participants to just like leave them outside the laboratory for the experiment. Make sure your design doesn't have any systematic flaws. This is EEG Basics 101, that you might generate spurious differences just by participating in the task, and then you might end up concluding that young versus elder participants are different, or patients' controls, etc. And make sure you have some sort of event marking procedure. Martin told us that beautifully, and we can definitely are convinced that you just can't just go and measure people interacting at a bar, because you need some sort of like event marking, okay? So, but that applies to also like basic EEG. So all of those go into the hyperscanning world. Pitfall number one, don't do it alone, team up. And those are actually the Power Rangers teaming up with the Ninja Turtles for sure. So team up, don't do it alone. These setups are really complicated to do. So if you were like a wizard doing your ERP task alone, setting up the EEG, you could do that for sure. But in the hyperscanning setup, they're it's double, literally it's double the work. So we actually, in the laboratory, we used to set up a common experiment with 2 people, usually a grad student and then an undergrad, a very well-trained undergrad, plus, I don't know, you could, our lab manager or something like that. Now we actually do double of that. We have more people there just to check everything. So team up and keep detailed records of everything regarding machines, software, setups. Since the complexity of these setups is so high, it's easy to actually just forget about details of what you did. And then when the next student comes, or even you, when you want to go and check your data, you might forget about something. You know, the follow-up study will need those detailed records. And have open science in your mind from the get-go. Recently, the EEG Beats was published. Cyril Pernet and collaborators made this very nice effort on trying to unify how to share data on the Beats. There are other languages or other options. This is just one. Do your research, because setting up your files is going to be complicated. You're not going to have only one file, but everything is going to be doubled for the EEG. Pitfall number 2, good idea, bad implementation. You know, Simon Ladouce and I are supposed to be writing this more deep, you know, wanderings into how to organize structured, semi-structured, and unstructured setup for hyperscanning and mobile implementations. But be careful with just going out in the real world without having clear hypotheses, because the more unstructured your experiment is, the more lack of interpretability of results you're going to get, and vice versa. The more control you will have, for sure, more interpretability, but also less ecological validity. So there's a sweet spot there. Simon and I have been talking about maybe doing this in an increasing way, just going to the real world and then going back to the blackboard if things are not working. So think your designs very well. Pitfall number 3, getting lost. There are lots of places to get lost. Get lost in cables. Martin already told you a little bit how to set up. You can find some articles. This is from Klaus's 2014 review paper, where he described LSL. And Martin already gave a really nice introduction into that. In the already postponed MOBI conference in June, there was going to be an LSL mini conference workshop. Everything is going to be moved to 2021. So if you're interested in that, anticipate that and try to go into the LSL workshop, because that's going to be very, very helpful. Organizing your files, I already mentioned that. It's not going to be trivial, as the volume of files is going to increase. The logistics are also going to get more complicated. And if you live in a country like mine, where basic technical skills are very low, in Chile, the technical skills are very low, you will find students that want to do really complicated tasks, really complicated design and experiments, but they are not managing the basic skills, you know? So basically, we are fighting against the technological usage, where nowadays most users of technology are just doing basic stuff. I am very hopeful that this COVID situation will actually push us into using technology in a better way. So this is something that I show my students. It's like, depending on the complexity of the phenomenon you want to study, and the use of systematic disciplinary work and the general and specific technical abilities, the line goes like that. So really, if you have complicated questions, prepare to put a lot of hours into studying and to getting to know your equipments and your literature, because this is not easy. This is very complicated. Pitfall number 4, analysis perpetuum. That is something that even with your ERP tasks, you might actually get into that. If you have been following the literature, there are 2 of my favorite books. One by Mayex Cohen, analyzing New York Times series data, and a recent one by Rita Hari and Aina Buse, MEG primer. If you take a look at those books, you will realize that what you can do with just a single channel of ERP paradigm data is amazing. It's a bunch of options that you have. And if you take a look at Arthur's recent review on hyperscanning, you will also realize that the options can be applied to hyperscanning. Here I modified the Arthur's figure just to add a connectome there, where you actually model everything in just one thing, because fMRI there was an fMRI figure there. So if you go to the paper, you will say, oh, it's not the same. I modified it. But the options are there, and the options actually of analysis are more complicated. Try to plan your analysis beforehand and have some very particular hypotheses. It's good to have hypotheses. It's good to go and test that. Of course, it doesn't forbid for you to then explore a little bit further. What we're doing in the lab, since we're actually researching on a very weird topic, which is psychotherapy, there's no time to go into that, but I can tell you about that later. We're doing parallel pipelines. So for example, we use Amica, which is like an algorithm, an ICA-based algorithm that has been around for a long time, and compare that. We're trying to compare with ASR by Bloom et al., which I know that is from Martin's group, Sarah Bloom, and she has a very nice implementation of this algorithm that is working very well. And I know that actually MBT's amplifiers also might have it at some point. So we are trying to hope for congruency. Applying these different things is a good it's a good exercise that we do in the lab, because every single student actually gets to do something with the data. And then even though it takes a little bit longer, we are very certain that these things are working. And also we're learning the different techniques that are around. And slow science is not a bad thing. Yeah, if we don't stop the whole publisher perish thing, I don't know who's going to do it. So it's better slow and certain. So summarizing, just to like go into the discussion section, know your systems, know your literature, partner with the company for sure, but also know your system. Try to hack it, try to know how it works, okay? Don't take it for granted, and know your literature. Nowadays with the internet, you can follow people on Twitter, you ask them questions about their paradigms. People are super nice, they reply to you. So know your literature. Plan data acquisition, storage, analysis as much as possible. Plan everything ahead, okay? Try to be a control freak in here. This is not bad. It's good actually to be a control freak in these situations. Team up and pilot as much as possible. Hopefully with a member of your lab or someone that you trust, because you don't want to, in an ethical way, you don't want to put participants in these situations, collect bogus data or collect data that you're not going to be using. So you want to respect participants' time and participants' privacy. So basically pilot will get dummy data or whatever, but team up, that's good. Make sure you always include meaningful behavioral or contextual measures. That means eye tracker, means a very good planned task. There are some cool things with smartphones on the streets, you know. Make sure you always include meaningful behavioral contextual measures. Try to reduce and control foreseeable artifacts, of course, that's EEG classic. Keep detailed records of everything. This is very important, because now that we have open science in mind, you want to give away this data and make people use it over there and maybe replicate you, of course. Make sure your results are not an artifact of your processing line, okay? So don't marry yourself maybe with just one analysis, you know, just regressing eye blinks or whatever. There are very cool toolboxes nowadays for this stuff. So get just your hands dirty on different analysis. Share your data and analysis pipelines with the world. Nobody knows what cognition looks like in like social cognition among different people. Might be high dimensionality, might be low dimensionality. We have no idea. So share your data, and let other people analyze it and compare, collaborate with people in the world. All right, so that's it for me. Let's go for discussion.
Okay, that was super interesting and very useful, and especially the point about making sure that your knowledge is actually in line with what you're trying to do. And I know that also the literature you named, that the Mike X. Cohen's book and Aina's book are really a good start there. In my personal experience, the complicated questions have very easy to understand, simple, wrong answers. So, but assuming we want to avoid that, yeah, you should really take one step at a time. All right, I'm really looking forward to continuing this with the questions from you guys. So I promised to ask some of the questions from the last time that your colleagues didn't have time to answer. So one that I'll start with is which are the main differences to pay attention to when preparing a multi-human study compared to individual human study. You can unmute yourself.
Go for it, Martin, please.
So, well, one of the larger problems is to get people to show up at the same time in the lab, which is really trivial. But indeed, if you have two people, the likelihood that one of them is not showing up at the appointment is rather high. So always call people beforehand, make sure that you have a phone number, and make sure that they know where they are supposed to be. So that is quite common a problem. This is really annoying because then you have to spend all the time, reserve the lab, have to pay one participant showing up, and then the second person is not there. So that's one of the major issues, actually.
And on the technical side, is there something to address specifically different than?
Well, the synchronization surely is the main problem. So, and depending on your level, as I mentioned before, depending on your level of precision you need, you have to really take good care that you are synchronized. And again, you need to test your systems. You need to feed what we always do. We play specific signals into both systems from which we know exactly how these signals look like. And then we check, do those signals arrive at the expected time? Do they stay synchronized over a long time? So normally we run with these artificial stimuli, we run an experiment for the entire time the actual experiment works, because it might work for the first 5 minutes, but after an hour it suddenly starts to break down because the hard drive suddenly is full and the computer starts to behave very oddly. So you are starting to handle quite a lot of data, and already this data assembling process might actually bring your hardware to a standstill, and therefore always check your entire experiment for the entire time. Also nasty problems in the beginning of your study, it works, and then after sometimes it suddenly does not work because the hard drive suddenly gets full, and it takes really a long time to debug. So make always sure that you have space available on your hard drive, and that you're really, really sure that your two systems are somehow synchronized with some objective measure. What we very often do is that we use the brain itself as a synchronization tool, that we play a sequence of sounds, beep tones. The N100 is quite reliable. And if you can see it in both systems at the same time, you at least have an indication that your systems are somehow synchronized. But better is to have technical solutions. And there are, from neurobehavioral systems, they have the lab streamer. So that's a device that allows you to detect the timing precision of your setup. So get some technical help to really know whether your signals are coming in on.
Yeah, I will add a little bit on because I agree with everything that Martin said. We usually prefer to, even though we use LSL, we usually prefer the basically hardware synchronization. There are some equipments where you can cascade them, basically make the EEG believe it's just recording a giant 128 or 256 channels, but actually it's just recording two separate files. That one for paradigms where people are just like kind of like doing something on computer, you know, all this like finger tapping task on synchronization. But then again, synchronization of equipment and knowing that the whole paradigm works is prime. So piloting, I would just stress on pilot, and make sure everything is up and running.
Yes.
All right.
I would like to add quickly on that, because if you have the possibility to do a hardware-based triggering and hardware-based synchronization, do that. In our work, we always want to do everything wireless. We want to go mobile. We want to go out of the lab. This is really, really difficult to do. So whenever you don't need to have mobile systems, you may get better results with hardware-based systems. On the other hand, wireless systems allow you to do so much more fun things, but then it becomes even more difficult. So I always speak about completely wireless systems, and then you need something like LSL testing
All right, this was the question actually from Hanna Poikonen, the one that you answered. And maybe just continuing in a similar way, I have a two-sided question. Is there a typical setting of hyperscanning in the sense of number of EEG channels, montages, dry or wet electrodes? And how do you make sure that the latency is low or controlled between data stream after the recording? So is there a way to know, let's say that you recorded something, whatever precautions you were taking before, is there a way to know that your streams are synchronized? And what about the hyperscanning setups?
Well, I can quickly show one way how we did that for one of our studies, or for the studies I was actually talking about. So because there we had a problem with the devices. We recorded from 2 mobile devices and both of them ran at 500 Hertz. Only later on we learned that 500 Hertz is not perfect because it leads to data loss in the streams. And I don't know whether you can see that here, but what we do a so-called ERP image. We take all the ERPs and we stack them on top. And now what you actually should see is then straight line across all your events. And what we see here in the second and also in the last plot is that there are sudden jumps in your data. So the signal started to drift and the ERP always came later, and then some correction kicked in and the ERP jumped back to the beginning. So this is a very easy way to synchronize, to check afterwards whether your data is plausible enough. Take some large ERP from which you know that it could be in your data, some visual evoked potential, some auditory evoked potential. And if they don't look good and they don't form a straight line, then at least be careful about how you interpret your data. That's the one way we try to fix that problem in our data set here.
All right. And what about the let's say, is there a standard of hyperscanning setup in terms of number of electrodes or montages or type of electrodes?
I mean, I would say that it depends, as always, as your research question, and your economical possibilities. More electrodes is more expensive usually. However, in terms of more movement, I think that you might want more electrodes in terms of movement, because basically, if you have very few electrodes and your paradigm involves moving around a lot or jumping, I'm thinking kind of like the Klaus's experiments, you know, you actually want to measure a bunch of other signals, including your noise signals. So characterizing your muscle, for example, signals, that would be highly recommendable. Hyperscanning in social settings, you don't need to actually just go and play volleyball to understand social interaction, you know, although it would be kind of like a really cool experiment, but you don't need to do that necessarily. You can actually think about your design and take it to a lab and do it very well. So in that case, I would say, just as always, try to go as high as possible. I think that nowadays with some modeling, that you can ask very interesting questions on your EEG signals. So more than 64, I would suggest, so you can actually have, you're not limited then just by a lack of electrodes. And I've been using dry electrodes from A&T lately. And although I haven't yet gotten an expert, you know, as an expert, just to get as good signal as with the wet electrodes, I think that dry or easy to put caps are going to be very crucial in the future of social interaction studies, because you don't want 2 people there and then put all the gooey thing, you know, in their heads. No, you might actually want to go real quick on your setups with high quality signals. And if you actually want to use children and a caregiver, which are very interesting dyads to understand, because they are, you know, let's say they are dyads that are way together, more dependent than adults or for example, clinical populations that are very interesting to study in terms of interaction, because I don't know, depressed patients or brain lesion patients, they have problems with social interaction. So studying those kind of things are going to be really, really important in the future. And having them sitting down for a 15 or 20 minute GU applying session is not going to be very nice. You know, so I would say that that's still one thing for manufacturers to improve their game in easy, quick setups with dry electrodes. I know there the war between electrodes companies, you know, it's still there. So developments are underway, but I think that's is going to be a very nice big step.
All right, we have a question. Yep.
Can I quickly comment on that? Because so I agree that it really depends on your research question. So if you want to do source modeling, you need a lot of channels. However, if you only are going to report one ERP of one channel in the end, and you recorded 128 channels, that might not be necessary. And for our mobile studies, it was really nice. We had 24 channels. We put the caps on. People were free to move. They behaved much more natural than what I observe when people have these heavy caps with a lot of electrodes. So if you need those electrodes, use them. If you don't need them and if you don't make any use of them, why should you bother with the extra time, with the extra cost, with the extra complexity? Because of course, with more channels comes more possibilities for problems. So really know what is your research question. Based on that, select your hardware.
Okay, that was useful. We have one question that is touching on a different point. You need a multidisciplinary team for all these studies because they're very complex. How do you manage such a team, especially if you're not the expert in one or the other topic? Question from Nizar Khawat. Previous one was from Luca Longo from TU Dublin, just to be transparent.
Let me get a go on this. So multidisciplinarity is the thing where you have to go. You know, like the centers, research centers, and laboratories have to go over there. There are different models in universities. You know, some universities give one professor, you know, one laboratory space, and they do their own thing, and they try not to promote a lot of collaboration. I've been in some departments like that. There are some other departments where you are more freely to collaborate and it's not so individualistic. In the center that I'm directing right now, we are 4 PIs with very different backgrounds. We actually have a clinician. We interact with psychotherapists as well, clinicians, you know, psychologists that are going to be doing that. We're very techie oriented, so we don't need engineers. We have enough friends, engineers, if we ever have problems, but we are very techie. We can program all our stuff. But sure, I would say that I cannot see nowadays a scientific effort that is looking forward in the next 50 years that is not multidisciplinary from the start. And even including having philosophers around is very good. If you have enough money to actually hire a philosopher, I always say the philosopher is someone, an experimental philosopher that has to be in your lab. That would be amazing. But yeah, multidisciplinary is a must for this kind of experiments or for this kind of setups. How do you manage that? Collaborating. So being open, try to get involved in the community. In the community, people are really open to actually collaborate, to lend their expertise. I haven't met anyone, a couple of people, but like in the last couple of years, let's say in the last 5 years, I haven't met anybody that had been like nasty or telling me like I won't share my data or anything like that. So I would just say try to open yourself to the community. If you work in a place where multidisciplinary is not so encouraged, or there are very few other people, try to go online. The whole world is there for your disposal, basically, and try to engage with other experts. I'm pretty sure Martin has something deeper to say.
I completely agree. So talk to each other simply and learn the language of the other disciplines. So it takes some time to understand what the actual disciplines mean, and be aware that you might not talk about the same stuff despite the fact that you're using the same words. This always takes some time. If you have a philosopher and a psychologist and an engineer, they talk in common day language, but each of the words might have a different meaning in the discipline. And it will take time to get to know these other languages, and you have to be open to it. And you have to accept that in principle, everyone wants to work together, but that you might sometimes simply fall into a communication trap. Make really explicit, are we talking about the right thing? How do you mean that? What do you mean by this? And be specific there, and talk to each other, and develop an error culture, so that you allow that everyone does not know anything, but that you can contribute everything. Everyone knows something, but simply be open.
All right, thank you very much. Now, I would like to make a small break for you two and ask our audience one question that is for our internal purposes, like just to know how good we tackled with these problems. So I'm just going to launch launch this poll and ask you to provide this as a sort of private feedback for us. The question is, have we helped to address the pain points of the hyperscanning process? All right, thank you very much. In case you wish to see, yeah, so you were very helpful for our audience. There are still a few questions, so we are going to get back to those questions. Actually, continue with them. I have a very technical one that Eina Pius from Indiana University and Laura Kaltwasser from Berlin asked last time, and it's concerned with sensor space analysis and measures of synchrony. So, what measures should we bring to minimize the confound of leakage? Or put in the other words is, how do you subtract activity due to the similar environmental input from emergent social matching activity? Do you have an answer to that? Francisco, you need to.
Oh, I was just saying, like, maybe Martin has a first take.
No, I actually don't have a good answer to that. So the problem was with all these studies, so if you share the same input, if both people hear the same sounds, then any kind of synchrony between the brains is in a way trivial, because you're exposed to the same sound, so therefore the brains will do something. Every ERP study is based on that. You present a sound, the brain reacts. If you present the same sound to two people at the same time, their brain will be synchronized. So in that sense, I don't really know yet how to disentangle these things.
Yeah, I also don't have a very good answer. I would just say try to extreme precautions. So the parallel processing pipelines. So for example, try that Sarah Bloom article on ASR has a very nice example on how no cleaning procedures, for example, and then she applies different algorithms and then compares at the end. I think that at the very beginning, at least, if of this field, we should be doing that kind of thing. And of course, sharing the data right away, so other people will implement other perspectives and other approaches, and then try to compare. You know, like I think that if we can do it from the theoretical point, we'll have to go empirical on that.
All right. Actually, Simon Ladus, who you mentioned, has a question for you guys. So, Francisco, you pointed out that there are many ways of analyzing EEG data, but are there specific analysis such as connectivity, maybe that seems particularly suited for investigating hyperscanning data?
Super good question, Simon. Hello, Simon. So I would say that it all depends on your question, you know, like what's the question you're trying to solve, and what is your epistemological perspective on that. So you might have, if you're very married to synchronization, for sure, there you have your answer. If your question is about actually how brains would actually synchronize at a moment, then you will have some questions about lag. Are you going to be doing zero lag synchronization? Are you going to be allowing for some lag? And that's a theoretical point. So basically, if I perform an action and someone looks at me performing their action and has some social meaning, is that information transmitted with zero lag? Should I be looking at zero lag synchronization between brains? Should I be using some sort of lag? That's an open question and an empirical one. And then if you might take another more radical approach, you might say, well, when you interact, you know, the only way to actually get meaningful interactions is longitudinally. So you will have to do some sort of like more than one session on your study, perhaps, and try to see how these things evolve. That would be like a very interesting way to see how you think of that paradigm. We use psychotherapy on that, so because basically we think psychotherapeutic environments are natural laboratories where people just talk and they have very established setups, so they last 45 minutes. They are sitting down, so there's not a lot of noise. There's very nice turn taking on the conversations, so you could do actually analysis on that. You can go into content analysis. You can do a bunch of other analysis that Susanna Decker and I are working on that project. But that's something that we came up because our idea is to actually try to see how these things evolve in time, like diachronically on this process. But if you have actual questions on events that might happen right there, you know, think of the Kelso-Tognoli experiments where people synchronize, you know, that's another kind of analysis that is completely different. So I would say that it depends on the setup you're trying to do, on the experiment you're trying to set up for your question. The analysis will be, I would say it will flow, hopefully, naturally, the kind of question and analysis you might be answering. Martin, I don't know if you want to add something.
Okay.
Beautiful.
Thank you very much. So I have a quick one from Konstantino Dragicevic. Do you consider the variable of knowing or not knowing each other? In the published studies, do interacting subjects know each other, or they are just first time?
In the studies we did, we decided that they should not know each other, but it was basically just a decision. We didn't do anything with this extra information. We just thought that it would be perhaps odd if people would know each other. Therefore, we decided for strangers, but we didn't systematically look into that.
All right.
I would say, again, your research question leads that. So if you're wondering about the development of a relationship, for sure they shouldn't know at the beginning and then do something covertly there. If you're going to add friendship or a companionship as a factor there, maybe in a problem-solving task, you might want to compare people that are known versus unknown. Again, your question drives that, I would say.
Yeah, that's probably one of the most important take-home messages here. So base your research on your research questions and not on the tool you happen to have at your with your hand. So you kept on repeating this, so I thought, repeat it once more.
Yeah. Okay, actually, we have a comment from the question from Konstantinov. Says, in fact, a longitudinal study would address the evolution from interacting with a novel subject to a known. All right, we have a couple of more technical questions. First was from Clemens Meithoff from Cologne. What are the current ideas or recommendations on setups involving dual EEG with simultaneous video recording? Do you have an answer for that?
Again?
So are there current recommendations on setups that involve dual EEG with simultaneous video recording in terms of either setup or equipment or best practices?
So we are doing we're doing a 2 EEG 4 camera setup for our psychotherapy setting, and the 4 cameras basically they are 4K HD USB cameras that they are plugged into a computer. That way LSL can actually just catch them as webcams. I suggest going high end for these cameras because you don't want to actually stream, and this is something I learned the hard way. You don't want to stream, of course, the video. You just want to stream the markers on LSL and just record the video locally. Otherwise, even if you have a dedicated Wi-Fi signal, it's going to collapse. So depending on your hardware, if you have a dedicated system per camera, so for example, a tablet or a phone or a laptop, see how many video streams can be recorded at once without losing frames or without even shutting down. I would say, and that's part of the piloting part. Okay, so if you're lucky and you have a super good computer, you can plug all your cameras in there and record with no problems. But you might find yourself with one system per camera recording the video locally and transmitting through LSL the markers and then putting them together. So in that sense, LSL will be the way to go. I think you might be able to do hardware synchronization, but I would say that at that point, it might be a little bit more complicated to find a camera that can be hackable enough, a commercial camera, you know, that is hackable enough to actually hardware synchronize it. So I would just go with LSL.
All right, thank you.
I think it's important to distribute it over different machines. So I think that's really a crucial aspect, that you build a local network, that all the computers are connected, and then that you run the heavy load on individual computers, that one computer is recording the video, some different computers doing the stimulus presentation, and the third one is acquiring your EEG. So distribute it over different computers, and LSL will do basically all this network operation. It will check how long the signal takes from one computer to the other. So it's quite convenient to run that on different machines. It runs basically out of the box most of the time, and it's much more stable than trying to run everything on one single machine, where you don't know how the different devices block each other and how the timing is. So I really second this opinion here.
Guillaume Dumas, who is an expert on this field, probably he should be here with us as well. He says that using TTLs is also a really good idea. Yes, I haven't implemented that. I've been using LSL, but for sure that TTL using is also a good idea.
Okay, a small comment from me. Yes, Guillaume Dumas will definitely be here in one of the next sessions. And he asks the question, some hyperscanning neural correlates are going up to the gamma frequency bands, so milliseconds, and thus go way beyond the behavioral time scale. So what are the underlying mechanisms according to you?
Martin?
I have difficulties to bring EEG and high gamma phenomenon into line with each other. So I don't know how far it's actually gamma that we are recording. So I come from ECOG recordings, so recordings in epilepsy patients. There I understand the concept of gamma. I don't know what you're recording with EEG in terms of high gamma. So I'm not really the person to go to with this question. So I'm very skeptical towards that, but without having really a lot of experience in trying to get this running.
Yeah, I will add on that my personal take. I'm also not a very big fan on gamma, mainly like gamma power. First of all, because I haven't worked much with it, so I don't have the first-hand experience on designing experiments where I'm going to actually get a really nice gamma effect on the EEG. So I would say that if your question is, if your research question and your paradigm is actually designed to trigger a gamma effect on the hyperscanning or mobile EEG, I would say get super good controls and be super, I don't know, obnoxious about it, you know, try to control it, try to destroy your effect as much as possible. And if it survives everything, I don't know, we might be getting into that. But there were some articles, I don't know, I think it was like the emotive system. I know that the emotive system is not the best equipment to have on your setups, but I think that they could only record up to 30 or 35 hertz. I don't know if anybody has done a systematic approach to mobile on MBT or ANT or whatever other brands, like a very systematic approach on what you are recording and how high and you actually get.
That's an excellent point. I think since we have only 5 or so minutes, we'll just try to jump to as many questions as we can. Just to repeat that we'll put this this talk on the YouTube channel, and there is also a Discord channel where you could actually engage directly in the talk. We'll share this as well. We have a question from Arvin Agrawal. The more naturalistic your experiments become, the more difficult it will be to analyze that data due to lack of clear or planned events. Francisco mentioned psychotherapy for 40 minutes. How do you analyze such no clear event data?
That's a super good question. Thank you for asking. So psychotherapy was chosen in my case because we think we can actually extract some very meaningful epochs or meaningful situations. So in psychotherapy research done by clinicians, and this is like a long tradition, people analyze the videos just like you're analyzing behavior, you know, like animal behavior. You know, they do the whiskering, you know, in the animal behavior world. They count behaviors and stuff like that. You can do exactly the same on the psychotherapy session, recorded session, with the goal of actually understanding what the interactions are. And once you understand the interactions, you can actually divide them and score them. They have developed all these manuals to actually do that and score them as good, let's say, good interactions and poor interactions in terms of therapeutical approaches. So I think they call them change and rupture, and they have at least 10 or 13 kinds of them. So if you think that way, you will be able to actually identify meaningful interaction periods during the psychotherapy where the content might be different, but the interactional settings or interactional conditions are going to be rather similar. That will be one thing. The other thing is that since you're recording voice and audio at the same time in high quality, you can use some words or some, you know, like vocal patterns, such as silences, such as changes in rhythms, changes in frequency in the voice, in order to kind of analyze that. Again, what Martin said, synchronization is the most important thing in those setups. And adding behaviorally relevant event is also important. So maybe just going out to the world and appreciate nature might not be the best necessary experiment to do. If you want to do something like that, you might want to attach some eye trackers and then try to do some fixation related analysis or stuff like that. But always add a behaviorally relevant event so you can actually try to interpret your EEG.
That was also very helpful. I have, I think I have more questions than the time, but let's just go straight to them. Given that the instrumentation protocols and pipelines are so hard for hyperscanning studies, should we kind of clearly define which questions only hyperscanning can answer? This is from Stephen Whitmarsh.
Go, Martin.
Well, as Francisco said several times, so you need to put yourself based on the research question. If you have a research question that can only be answered with hyperscanning, then go for it, and then take the extra effort to get this system up and running. If in the end it turns out that you don't need the complex setup, well, then it may have been a waste of time. So really always ask yourself, despite the fact that you have so many nice hardware possibilities in your lab, do I really need them, or is actually a button press experiment sufficient? So as an EEG lab, you always have the problem, yeah, we are an EEG lab, so therefore we are going to use EEG to answer this question. Sometimes it turns out we could have simply asked people or let them press a button. So really see, do I need this method to answer these questions, or am I just using this method because I know it and therefore somehow tune my questions towards this? Really let yourself guide by your research question and then check what's the best tool, and then you may want to take the extra effort to collaborate with someone who actually has the technological means to answer your question, but don't let your research be completely guided by the method, unless you are interested in the method.
All right, thank you very much. Quick one. Nowadays, some EEG systems works with USB-based devices. In context of this story, is this a problem for time accuracy? Martin, what do you think?
No, I don't think so. So again, if the manufacturer is doing things right, you can screw up on any kind of hardware connection. I think there are a couple of companies, good EEG companies, that use USB devices, and I'm not aware of any problem there.
Okay.
I would say,
for example, I know that some sound cards that come with USB plugs, they might introduce some lags which are not good for your setups, but for EEG, I'm not aware of any problem. You might want to add.
No, I was just going to say, I agree with you. I agree that I haven't heard any complaints about using USB. I would say it's more dangerous to actually hack your system poorly. So I've seen some people like attaching an Arduino to the amplifiers just in order to be able to put a parallel port. When sometimes they are working with Macs or stuff like that, that they don't carry parallel ports anymore. So I would say that poor homemade solutions are going to be definitely worse than the manufacturer's provided USB, even if you are a little bit suspicious about USB. But I haven't heard anybody complaining.
And also make sure, no matter what system you use, that you characterize your entire system and the temporal behavior of the entire system. Also, test as soon as possible in the lab you're going to measure in the end. It might be the case that on your local desktop computer it works with your microphone, and then your entire paradigm works, your timing tests are fine, then you move to the lab to a different version of Windows, to a different version of MATLAB, or some sound card properties, and suddenly everything is off. So don't spend timing tests on your local machine, but always test on the final machine you're going to record with all devices set up and all devices already activated. And only then you can get a good characterization of the temporal structure of your setup and place and everything. That's
Okay.
also something we learned the hard way. So we developed things beautifully on our local machines. We moved to the lab and we started all over again.
Last question. How will the extraneous variables would behave when recording is done outside of lab settings? For instance, mood, anxiety in a simple working memory task.
I didn't get the last part.
If you have effects of the outside settings that affect the mood or anxiety or something, how that would affect, let's say, working memory task as opposed to inside of lab settings?
That is simply a great research question you can start to approach. So these are things we don't know. We know that movement and going outdoors has influence on creativity, on all kinds of things, but we actually know, I think, quite little about this in the context of brain recordings. So there's a question you can start to tackle with what you learned today.
Okay, well,
yeah, so quickly, that question is actually a very 4E question epistemologically, because you're asking about like how a context where your cognitive ability, which in this case is memory, is going to be embedded in some maybe exogenous background or anything like that. And that's for sure like some very exciting ways to go. So yeah, thank you for that.
And in classical studies, we always try to get rid of these factors. And now we suddenly face those factors and we are trained to get rid of those factors. So we really don't know how to handle it. And we get anxiety because we say, but I can't control it. But actually, the realistic context is not controlled. So somehow you have to deal with it and somehow you have to embrace this complexity. And I think there's no definite answer yet how to do that. It makes this entire field much more useful to
just to respect the person who asked the question is Prashant Saha from GFSU in India. I actually have more questions, but I think I kept you for a long time, and for me it was a great pleasure talking to you, and it was really useful hearing your views and hearing about the issues, and hearing that you should not trust your producer. That actually gives me hope. But anyway, I would agree on with most of what you said about checking the setup, about the needs to be able to set up your experiment in a fast way, to check everything, to make sure that your level of knowledge matches your research paradigms challenges, and all the technical aspects that we need to make sure of. So again, Martin and Francisco, it was really great having you, and I hope we keep chatting on Discord or mail or Twitter, and that you get out of maybe not lab, but get out of your homes soon. That's my wish to you.
Thank you, Ivan.
Thank you very much.
Okay, this concludes it. We'll share these videos, and see you next time.
Bye-bye.
Bye-bye.